Well, good day, my fellow colleagues, healthcare practitioners. What we'd like to cover today is some discussion on a new field called epigenetics and how epigenetics affects allergy practice and why we should know about it. So the objectives of this presentation are to recognize the importance and relationships of epigenetics to the immune system in health and disease, understand the molecular and epigenetic mechanisms regulating the immune response, and appreciate the future directions of this field of epigenetics and its clinical application to allergy practice. These are some of the topics we would like to cover. The first is the overview of allergy, the immune system, and epigenetics. Allergic diseases, by definition, are a group of chronic inflammatory diseases that display a broad spectrum of clinical manifestations and conditions and are primarily mediated by IgE, but not exclusively. And these are commonly referred to as the atopic disorders. And these include allergic bronchial asthma, allergic rhinitis, allergic conjunctivitis, atopic dermatitis, and food allergies. Now there's a concept that we're all familiar with, which is called the atopic or the allergic march. And although each patient shows an individual spectrum of these conditions, certain patterns are well recognized during the lifetime that we refer to as the atopic or the allergic march. And this is defined as the sequential manifestations or evolution of allergic disease at different ages, beginning in infancy and childhood, as atopic eczema or food allergies, and shifting then later in life to respiratory conditions during which compromised barrier function leads to other dysfunctional epithelial barriers. And here in this slide, we see the progression of the atopic march, beginning at birth, at 5 years, at 10 years, and at 15 years. And we see eczema with this red line being one of the first manifestations in the young infant, followed by food allergy, asthma with this blue line occurring in 2 or 3 years of age, maximizing at 5. And then rhinitis, allergic rhinitis, beginning at about the same time, and coming up a little bit later. So there is a progression of different disease expressions at different ages that is reflected by the atopic or allergic march. Now, there's another concept that we should cover, which is called the hygiene hypothesis. And it begins with strong epidemiologic evidence demonstrating that there is a steady rise in the incidence of allergic and autoimmune diseases that has occurred in developed countries over the past several decades. And there have been several papers, and the one that highlights this most graphically is the paper by Bach in 2002 in the New England Journal. And this increased incidence has included asthma, allergic rhinitis, atopic dermatitis, representing the allergic diseases, and the multiple sclerosis, type 1 diabetes mellitus, and Crohn's disease, representing the autoimmune diseases. And this increase has been attributed to a decreased exposure to infectious diseases through immunization and improved sanitation. And this has been highlighted by papers by Strachan, 1989, in the British Medical Journal. And it's referred to as the hygiene hypothesis. And in this paper by Bach from the New England Journal, we see two panels here that are contra positioned. Here we see a decrease in many diseases from 1950 to 2000, such as rheumatic fever caused by the beta hemolytic strep group A, hepatitis A, measles, mumps, and tuberculosis, related to vaccine usage, cleaner environments, and antibiotics. But at the same time, we see a curious increase in many of these autoimmune diseases. We see an increase in multiple sclerosis, Crohn's disease, type 1 diabetes, and asthma occurring between 1950 and 2000. Now, just as an overview, for ease of discussion, we can speak about the immune system in health and disease in three compartments. The three compartments are the innate immune system, the adaptive immune system, and the tissue-damaging parts of the immune system. And the immune system consists of this set of cells and cell products that recognize foreignness. And if we look below the figure, if foreignness is encountered first by the innate system, this is the system, the older system we have, present at birth, we can use cells that ingest foreign particles by phagocytosis, such as the neutrophils and the macrophages, and other cells such as mediator cells, such as the basophils and mast cells that give rise to mediators, natural killer cells, inflammation, all are part of the innate system, together with a system called the complement system that augments the system. If foreignness, such as inert materials, infectious materials, or malignant cells, are recognized and eliminated, that's the end of the story. But most of the things we encounter are more complex, and they involve a second component of the immune system, the adaptive immune system. And here we have two populations, the T cells and the B cells. The T cells, we'll talk about, have a number of daughter cells called Th1, Th17, Th2, and Th3 and Tr1. These are called the T helper cells. Another group of T cells are the cytotoxic cells that defend us against viral infection and cancer. The other cell population that's important are the B cells that make antibody. And each of these colored molecules here represent the various classes of immunoglobulins. The big ones are called IgM, the lavender are IgG, the IgD are in yellow, IgA is in yellow, and the IgE are in red. Now, again, if foreignness can be eliminated, such as viruses, bacteria, fungi, or malignant cells, by this system, with the help of the innate system, we consider this the immune system in health. And we color this blue because that's a very peaceful color, and it means good things, so that this is a good outcome of the immune system response in health. However, if foreignness cannot be eliminated, we enter the third compartment. And this is the tissue-damaging part of the immune system, where the immune system is no longer beneficial, but here it's detrimental. And we have a system called the Jelen-Coombs system, where we have 4 types of reactions, type 1, 2, 3, and 4. Type 1 is where IgE-mediated disease occurs with these red molecules, the IgE. Type 2 are the cytotoxic, where the lavender and the green IgG and IgM can cause cell death. And we also have type 3, where immune complexes of antigen, antibody, and complement can damage tissues, and a type 4 where the T cells can give rise to damage through a delayed hypersensitivity or cell-mediated immunity. So if we enter this compartment where antigen or foreignness cannot be eliminated, we see the clinical expressions of allergy, autoimmunity, chronic infections, and malignancy, where there is failure of elimination of foreignness. Now, if we can eliminate foreignness, we get back to the blue zone in certain diseases. For example, if a patient has an allergy to penicillin with a rash, these patients will have a rash only as long as the patient is taking the drug. When we eliminate the drug, the rash disappears, and we return to a system of health, of the immune system in health, with elimination of the foreignness. In certain diseases, such as autoimmunity and malignancy, where the foreignness cannot be eliminated, the phase of tissue damage is more protracted. So without elimination, we see a continued damage. With elimination, successful elimination, we see a reversion back to health. Now, the immune system has many barriers that involve natural skin and mucosal barriers and the target tissues. And they're all interrelated and interdependent. Over here, when we encounter something foreignness that enters the body, the natural barriers are the gastrointestinal tract, the skin, and the respiratory tract. These are where foreign agents usually come into the body. And the first system that, this system helps keep out many foreign configurations, but some do get through. And the first system that's involved is the innate immune system of the phagocytes and the mediator cells and the inflammation, the complement system. And then this system, the innate system, interacts with the adaptive system, where we see the certain of these cells are called dendritic cells, process antigen and give the peptides to the T cell and the B cell. And then the fourth area is the target tissues, which can sustain damage, which are similar to the portal of entry, the skin, the mucosal systems, and the tissues, cells such as fibroblasts or smooth muscles. And this gives rise to disease, which can be chronic or acute, depending on elimination. And we show here tissue remodeling. If the antigen or the foreignness cannot be eliminated, sometimes these changes in these organs are irreversible, and we refer to that as tissue remodeling. So, common to all of this story is the fact that chronic inflammation at body environmental interfaces involving the GI, the respiratory, the skin, the mucosal membranes are important. And this reflects an active, chronic, misguided immune response directed against one's own tissues. And substances that cause allergy are referred to as allergens that lead to this immunologic imbalance. Now, the concept of balance is extremely important. If we have a balanced system, we are in a state of immunologic equilibrium. It's like in physics. Every action must be countered by a reaction, an opposite and equal force in a reaction. So when we're confronted with foreignness, pro-inflammatory cytokines are produced that are counterbalanced by anti-inflammatory cytokines, as in the case of bacteria in the gastrointestinal tract, which stimulate some of these cytokines. So they're equally balanced, and this is a system of health. When the system is imbalanced, when the pro-inflammatory cytokines cannot be counterbalanced by the anti-inflammatory cytokines, we have this system of immunologic disequilibrium, which causes disease. Now, we're going to focus later on in this lecture on some of the T cells that produce the anti-inflammatory cytokines, and these cells are called T regulatory cells, or Treg cells. And they make certain cytokines, such as TGF-beta and IL-10, that are important in this anti-inflammatory cytokine cascade. So the type of responses that we see in allergy are referred to as allergic type 2 inflammation, as opposed to type 1. This imbalanced immune response leads to allergic inflammation with the development of these T helper cells, or Th2 cells, as the consequence of allergen presentation by the antigen-presenting cells of the innate cells of the system. Following the exchange of a set of signaling events, these naive T cells develop into Th2 cells, which are defined and characterized by the type 2 cytokines, which include IL-4, 5, and 13. To show this more graphically, if we divide the system into those that affect the C, the effector helper, the effector T cells, antigen is presented to the antigen-presenting cells. The peptide then is presented to the cytotoxic, we refer to these as CD8 cells. These expand with cytokines into daughter cells that are called cytotoxic cells. And these are involved in cytotoxicity that kill viral infected cells and cancer. On the other side, when antigen is presented through an exogenous pathway to antigen-presenting cells, we invoke the T helper cells. The T helper cells are designated CD4 positive. These expand under the influence of cytokines such as IL-2. This expands into a daughter cell called Th1, CD4 helper cell, which makes its own type of cytokines such as TNF and interferon gamma. A second daughter cell is the Th17, which also makes its own cytokines, 17, 21, and 22. And these are referred to as the pro-inflammatory. These promote inflammation, pro-inflammatory cytokines. On the other side, we have the development of another daughter cell, the Th2 cell, which makes its own cells, cytokines, such as 4, 5, 6, 10, and 13. Another development of a daughter cells are the Treg cells that we spoke about. These are sometimes referred to as Th3 or Tr1, and they make the anti-inflammatory TGF-beta and IL-10 that we spoke about earlier. So these are the regulatory cytokines that are made by the Treg cells, and the helper cytokines by the Th2. These are the ones that give rise to allergic disease. And they do so because the Th2 system interacts with the B system to make antibody, and by differentiation of the B lymphocytes into a plasma cell that makes all of the protective antibodies, GM, GAM, and E. But sometimes the E is the one that gives rise, when it's made in increased quantity, to allergic disease. And showing it more statically, we have the endogenous pathway with the CD8s and the exogenous pathway with the T helper population in the producing all of the cytokines and interactions that we spoke about. Also included in this is another important receptor called the CTLA4, which gives a inhibitory signal, and in recent years, cancer immunotherapy has relied on stimulating this particular inhibitory molecule to enhance the immune system in immunotherapy of cancer. And these are called checkpoint inhibitors. So allergic type 2 inflammation is shown here with the antigen-presenting cells, the naive T cells, the Th2 cells, the B cells, affecting the various target cells of epithelial cells, eosinophils, epithelial barriers, and disease. All mediated, in this case, largely by IgE. Now, having said that these are the mechanisms of either protection or injury, we enter a field referred to as epigenetics and clinical disease. And this began with the sequencing of the human genome at the turn of the century, which heralded the exciting possibility that all diseases could be understood by looking at DNA. This week, I was reading a report from Dr. Francis Collins at the National Institutes of Health, who is the director of the institutes. And the NIH is now beginning in 2018, will be recruiting subjects, to donate their DNA to develop a data bank that will make associations between genes and disease. And this is an exciting project of the NIH, and it involves all of the great advances that we've learned about the DNA and how we have sequenced the DNA and have learned how that molecule is constructed and how it contributes to disease or immunity. So this is part of the genome-wide association studies, abbreviated GWAS, which originally failed to fulfill this promise. Now, attention is turning to the idea that mechanisms by which the book of DNA is read and not the book itself might hold the answers, and this comes into the field of epigenetics. So what is epigenetics? So the derivation of the term epigenetics comes from the Greek. It means above, epi, the gene, genetics. And this was largely contributed by the work of Conrad Waddington, a leading British embryologist and geneticist, who was the first to coin this term to describe the processes by which the genotype brings about the phenotype. So epigenetics has received a facelift in the modern era to encompass mechanisms that control heritable changes in gene expression not caused by changes in the DNA sequence. If we look at the DNA sequence as a necklace, it's made up of 4 beads, A, T, C, and G, adenine, thymine, cytosine, and guanine. And this is the man who really introduced this term, and he was referred to sometimes as the last Renaissance biologist. So epigenetic regulation is a mechanism that allows the genome to integrate intrinsic signals with environmental signals. If you look at this whole story of genes and epigenetics, the genes are the gun, and epigenetics are the trigger. So you can't have the gun without shooting it unless somebody pulls the trigger. And the trigger is involving the environmental signals that control epigenetics. So let's talk about some of the molecular signaling events that cause all this to happen. For ease of discussion, we get these complex terms that are sometimes confusing, and it's helpful just to simply define them, and we lead to a better understanding. If we look at a cell made up of a nucleus, which contains the double-stranded DNA, and cells all have receptors on them that respond to certain external environmental substances that we call ligands. That means something that binds to a receptor. We can talk about the 3 T's. And the 3 T's are transduction, transcription, and translation. The first T, transduction, occurs when the ligand binds to the receptor and sets up a set of biochemical reactions that involve phosphorylation. And phosphorylation is conducted by enzymes that are called kinases. And here we see JAK. JAK is a type of kinase, Janus kinase, that catalyzes the phosphorylation of a phosphate on a protein that here is called STAT, and we'll come to that in a minute. Once the protein is phosphorylated, it's activated, and it activates another protein, and so forth. It's like a cascade. These molecules are called transcription factors that come to the nucleus. Now, this first phase is referred to as transduction, which comes from the Latin transducere, means to lead across in Latin. And what is being led across is a signal determined by biochemical events that involve phosphorylation, kinases, that have a variety of names, that end up with transcription factors that come to the nucleus that set up the second T, which is referred to as transcription. These molecules that are phosphorylated enter the nucleus. They sit on a region of the double-stranded DNA referred to as the promoter region and initiate transcription, which comes from the Latin transcrivere, which means to write across. And what is being written across is the code of the DNA into RNA. And this is mRNA in red here, which is referred to as messenger RNA. And we'll come to that sequence and codes in the next slide. But once the messenger RNA is produced, it leaves the nucleus and comes into the endoplasmic reticulum, which is the protein factory of the cell. And the messenger RNA sits along these little ribosomes, which are the protein factories. It acts as a blueprint, and it ends up lining up amino acids by its code, and this produces a protein, which is then referred to as translation, which comes from the Latin to carry across. So when you hear these terms, transduction, transcription, and translation, you will have a better idea of how they function. Now, these then are the molecular signaling events of the 3 T's. Now let's move to the mechanisms of epigenetics. The way that the message of DNA is transferred to messenger RNA, this transcription, the second T, is the code of the necklace of DNA is specified by those 4 beads. A, C, T, and G. Whenever you have a T in the DNA, it links to an A in the messenger RNA. An A in the DNA links to a uridine. We change here from a T to a U in that situation. C, the cytosine with a guanine, G with a C, and so forth. So you're getting a mirror image of the messenger RNA based upon the code of these nucleotides of the DNA. And that's how the messenger RNA is produced in transcription from its gene expression. And we don't have to get into exons and introns and exons. Those are just pieces of the DNA molecule. And that leads to the production of a protein, which is referred to as translation. So we've got transduction, transcription, and translation. Now, the mechanisms of epigenetics, as we mentioned, the DNA is made up of a necklace of those 4 nucleotides. And in contrast to prokaryocytes, these are bacteria, eukaryocytes, which are the higher forms, the human, these have nuclei with more genes and more DNA, in which the DNA is packaged as chromatin. In bacteria, the DNA is circular. It's a single circle. But in the higher forms, we have them in discrete chromosomal packages. And there is a chromatin subunit which consists of nucleosomes. If you look at the DNA, it comes with 2 loops around an individual protein called a histone. And then the DNA comes to another molecule of histone with 2 loops and so forth. Each of these units, the double-stranded DNA with the 2 loops and the histone, consist of a nucleosome. And this is another nucleosome and so forth. When you have all of these nucleosomes, it's referred to as chromatin. Now, we're going to be talking about how that structure of DNA or the histone can be modified to give epigenetic changes that control the expression of DNA. The histone is sometimes acetylated with this AC. The DNA can be acetylated, but more commonly is methylated with this CH3 group. And when the molecule is modified, it has a different expression. So nucleosomes are then organized into chromatin that these not only serve to package DNA, but also to regulate accessibility of the DNA to the transcription molecules. And we'll talk mostly about DNA methylation, and these are important. They serve as signals to chromatin, modifying their expression. So let's look at the basic molecules. The best studied epigenetic mechanism is DNA methylation, and this little ring structure here represents one of the nucleotides referred to as cytosine. And when it's methylated, a CH3 is added to the number 5 position. It's like a benzene ring here. There's 6 carbons, but the 5th one is methylated by DNA methyltransferases. These are enzymes that put a methyl group on the cytosine to convert it from cytosine to 5-methylcytosine. And a lot of this is conducted by enzymes that are modified by vitamins and folic acid that are important in this whole story. Okay, the major epigenetic mechanisms include are 3. the DNA methylation, histone acetylation and methylation, and RNA gene silencing. And these serve as signals to modify chromatin complexes into transcriptionally repressive or permissive configurations. Now, we see a DNA double-stranded molecule here. Nucleosomes are the beads on the string, and we have two forms of chromatin. One is called Euchromatin, where the nucleosomes are separated, Heterochromatin, when the nucleosomes are compacted. And here we see the Euchromatin with the nucleosomes spaced, and the Heterochromatin where the nucleosomes are packed. And as you can imagine, the three T's, in order to achieve transcription from transduction, those molecules have to come into these open positions. So the Euchromatin form of the DNA is the only one that's operationally effective in being modified. Heterochromatin is non-functional. So when the, here's the Euchromatin in the open position, this is the active gene, Euchromatin, and here's the silenced gene, Heterochromatin. And they're all affected by these enzymes that transfer, in the case of DNA methylation, the methyl group onto the DNA molecule. Now, how does epigenetics come into disease? Well, the best studied role of epigenetics are with autoimmune diseases. And the one that was best studied, has been best studied, is the disease called systemic lupus erythematosus that we're all familiar with, SLE. Here's a young woman with fever, a facial rash, sometimes referred to as the butterfly rash, a joint pain, all caused by that third phase of the immune system, the red zone, and by one of those mechanisms that we spoke about, immune complexes, or sometimes by direct cytotoxic effects. It involves the kidney, and many of these patients suffer from kidney involvement with passage of red cells in the urine, hematuria. And you could see the changes by light microscopy or by electron microscopy. And they show these electron-dense deposits adjacent to the glomerular basement membrane, which affects renal function. Now, how did this story of epigenetics occur? It occurred by taking careful histories. And workers studied monozygotic twins and dizygotic twins. In other words, these are identical twins that emanated from the fertilization of a single ovum, and they're genetically identical. And if these diseases, if lupus was to be transmitted only by genetics, you would expect 100% concordance. That is, if one twin developed lupus, 100% of her identical sister would get the disease. But it turns out that it doesn't work that way. Only 12 to 67% involvement of twins. And this suggested the involvement that other things besides the structure of the necklace, the beads, was controlling whether that gene was open or closed, whether it was operative or silenced. And a role for environmental factors evolved, which strongly suggests that certain drugs, ultraviolet light, diet, can act upon the genetically susceptible subject, causing the gene to either be open or closed. And this is how epigenetics entered medicine. So DNA methylation involves systemic lupus, rheumatoid arthritis, multiple sclerosis, type 1 diabetes, and this is under DNA methylation, but RNA mechanisms are also involved, and histone. But we're going to concentrate on the DNA methylation story. So this paper, which came out in 2007, showed that DNA hypomethylation is crucial for apoptotic DNA to induce systemic lupus in a mouse model. And the systemic lupus is characterized in the human as well by antibodies against anti-double-stranded DNA. We refer to that as dsDNA antibodies, and we use this diagnostically. Apoptotic DNA is hypomethylated, while necrotic DNA is methylated. The apoptotic DNA is able to induce the antibodies in this model of lupus. And these simply show the normal DNA and the apoptotic DNA show different properties in terms of inflammation. So the DNA hypermethylation versus DNA hypomethylation refers to whether it is a closed switch or an open switch. DNA methylation is an epigenetic modification that influences gene expression. In SLE, there's a genetic hypomethylation of DNA, and many cells of the innate and adaptive systems that control inflammatory responses are open. So when you have hypomethylation, the switch is open and it's producing all of these anti-inflammatory cytokines that give rise to the symptoms. Therefore, the switch is open, resulting in excessive pro-inflammatory activity. Now, since DNA methylation is typically an inhibitor of gene expression, lupus-related hypomethylation around genes that control these cytokines are produced that give rise to these inflammatory conditions. And this leads also to elevated levels of interferon alpha, which is one of the cytokines that has been measured in this particular disorder. Now, we move from epigenetics and autoimmune disease to epigenetics and cancer. Epigenetic alterations, including DNA methylation, histone modifications, and RNA expression, have been reported in cancer. The carcinogenic process is driven by the accumulation of genetic and epigenetic alterations that result in dysregulation of key oncogenes, tumor suppressor genes, and DNA repair housekeeping genes. The administration of hypermethylated nucleotide sequences we have recently found stimulate Treg cells. The stimulation of Treg cells could offer a therapeutically beneficial effect through their known anti-inflammatory effects, and these observations offer the possibility of developing a vaccine for these disorders. But let's go back to the story with cancer. In the case of cancer, the zygote that's produced that leads to an adult gives rise to either normal epithelium or invasive carcinoma. And what occurs during developmental biology is sometimes the switch is open to allow differentiation of cells and organs, but at certain times they have to be closed, and that's when the DNA is methylated. But in cancer, if you have unmethylated DNA in the oncogene region, the switch is open, and that's not a very good thing. In the tumor suppressor activation, the methylated DNA is closed. It is not very good. So we want to have the tumor suppressor activation open, oncogene activation closed. So in cancer, the oncogene is activated. Normally, the oncogene should be closed. It should be methylated. But due to the malignant transformation, when it's unmethylated, the switch is open, and this leads to oncogene activation. The tumor suppressor region should normally be open. That means the DNA should be unmethylated. But in the malignant transformation, it's methylated, and the switch is closed. So these are the two events that occur that lead to the activation of cancer. And several papers have been written about epigenetics of lung cancer. Here's a paper from 2015 describing epigenetics in lung cancer, where the group of enzymes that catalyze the attachment of the methyl group are upregulated in lung cancer. That means it's closing the switch. Histone deacetylases, the enzymes that remove acetyl groups, allowing histones to wrap the DNA more tightly, are also overexpressed. So there are ways that this knowledge can be extremely helpful, because there are inhibitors of the enzymes that transfer methyltransferases, and they're also inhibitors of the molecules that are involved in acetylation. So these offer potential means for therapy. So in cancer, it results from the accumulation of genes called oncogenes and or tumor suppressor genes, with loss of function of those and gain of function of the oncogenes. So this has highlighted the importance of epigenetics in cancer. Now let's end by its applications of epigenetics in allergic disease. Epigenetics and allergy, from basic mechanisms to clinical applications, is a paper by Podajek here in Epigenomics 2017, which describe a hot topic for allergic disease. And papers have been written in atopic dermatitis, asthma, immunotherapy, the gut microbiome, and smoking that cite certain findings that major biologic pathways represented, for example, with atopic dermatitis, involve these mechanisms of epigenetics. And in asthma, the ability of these studies to link susceptibility has been published. And this goes with the microbiome, which is the gut flora, which is connected to diet, nutrition, probiotics, and other lifestyle variables also affect epigenetics, as does smoking. So of the major T helper cells, their differentiation and their epigenetic regulation, we find that there are crucial allergy-related functions. And here's the Th2 population. Here is the Th17 population. Here is a Th9 population, a Th1, and the Treg functions, all controlled by these molecular events. Now, the clinical applications in food allergy, there are a number of population-based studies, mostly focused on DNA methylation. And smoking influences the expression and methylation of receptors involved the innate system. Immunotherapy, epigenetic biomarkers are a surrogate for the effectiveness of treatment. With FOXP3, this is a molecule expressed only by the Treg cells. So we see clinical applications of epigenetics involving diet, such as folic acid, which is a source of methyl groups, fish oils, stress, obesity, pets. Here we see a pregnant lady and the baby, all involving epigenetic modifications of DNA, either by histone modification, DNA methylation, or microRNA involvement. The effects of smoking, air pollution, farming. This gets us into the microbial, epigenetic story that that led us into this discussion. So epigenetics is important in zygote differentiation. Remember, the DNA has to be open for organ development and closed at certain points, going from fetal development to adulthood. You know, there is a common disease that we used to see in babies, which involves the defects of the spinal cord with meningomyelocele. And we now give folic acid to pregnant ladies, which is a source of methyl groups that has prevented these things from occurring. So there are some clinical applications that have arisen from these studies. Likewise, cigarette smoking is a very potent epigenetic modifier, and the elimination or prevention of cigarette smoking has led to a lot of disease prevention, such as cancer. Vitamin C also affects methylation, as does smoking, and these are studies performed in pregnancy. So the future of epigenetics, as I mentioned in the introduction, genetics studies involve the gun. The epigenetics involves the trigger. And if we could learn more about the environmental causes of why the gun is triggered, this should offer new approaches to therapy and a better management of these diseases. Now, one of the original observations that played into this story are some observations by a surgical colleague in the early 1900s by the name of Coley. In New York, he found that patients with cancer, certain forms of cancer, who developed an infection, the cancer was ameliorated. And he spent his whole life looking at Coley toxins. He was growing bacteria to get these extracts to treat patients. It turned out that the reason why these infections were effective is that they led to certain forms of DNA referred to as CPGs. This is the cytosine guanine. These are regions of the DNA that we call CPG motifs. And a doctor later by the name of Krieg found that mammalian DNA tends to be methylated. Bacterial DNA tends to be unmethylated. Now, it turns out that if you inject methylated DNA into a host, you don't get an immune response. The immune response is turned off. If you inject unmethylated, the immune response is turned on and promotes inflammation. So taking these observations, these CPGs are modified to bind to some molecules on cells which are called toll-like receptors. TLR, and there are about 10 of these in the human. The ones that are binding the DNA bind to toll-like receptor 9, which is here in the cell. The DNA that is released from cells undergoing apoptosis is methylated and leads to no inflammation. The DNA released from cells undergoing necrosis is unmethylated and leads to increased inflammation. A cell, shown in this diagram, can either die by apoptosis, autophagy, oncosis, or pyrotosis. There's a whole spectrum. These responses of apoptosis lead to non-inflammatory cell death. Pyrotosis and necrosis lead to forms of DNA that is unmethylated that lead to inflammation. And several papers have appeared suggesting the use of these oligonucleotides, these CpGs, for treating patients. And one of the applications involves the use of unmethylated DNA with immunotherapy with ragweed. And the studies of Dr. Kretikos, the New England Journal, coupled a unmethylated CpG with a vaccine with the ragweed that showed that it could augment the immune response. So we've taken a different view. We want to take DNA, CpG, that is methylated. In a recently, some studies we performed, which will appear recently in the Allergy Asthma Proceeding March issue in 2018, we compared normal subjects, patients with SLE, and normal control subjects, stimulated either with the DNA as a stimulating agent or PHA. These are studies done in what are called lymphoproliferative assays, and where we measure the incorporation of tritiated thymidine into the cell. And when the numbers get bigger, this means the effect of the agent is positive. So with PHA, which is a plant mitogen, we got a stimulation index of 260, but using normal DNA from the human or from patients with SLE, the stimulation index was lowered. In these studies, we synthesized 5 sequences consisting of 8 nucleotides. They're referred to as octamers. And in 3 of these, we placed a methyl group. In sequence 1, the methyl group was placed on the 3rd cytosine. In sequence 2, the methyl groups were placed on the 1st and the 3rd. And in sequence 3, it was a different backbone structure. It was placed on the 1st. The sequence 4 and 5 were unmethylated and served as methodologic controls. Using sequence 1, which was the methylated, and the DNA 4, which was the unmethylated, we had an increase of the Treg cells measured by FOXP3 in the DNA which was methylated, which was greater than those obtained by the unmethylated. And there was a dose-response relationship that as we increased the concentration of the methylated form from 0 to 100, we increased the amount of stimulation of Treg cells. So this offers a very interesting possibility for cancer, for autoimmune diseases, and for allergy. And one of the areas that we're particularly interested in is taking our in vitro findings and seeing if we could extrapolate those into in vivo findings, that is, to use the methylated form of the molecule as a vaccine to stimulate the Tregs that would treat allergy and autoimmune disorders where there is a deficiency of Tregs. So we end with the same story that we began with of the immune system, of consisting of innate, adaptive, and tissue-damaging effects, and how epigenetics and genetics plays a role in this story. The allergic pathway is exquisitely regulated by epigenetic mechanisms. And we spoke about DNA methylation primarily, but there is histone modifications in RNA that are also involved in epigenetics. They play an important role in understanding allergy and immunotherapy. This knowledge could also be used to identify new and effective therapies or diagnostically as looking for biomarkers. And it holds the key to unraveling many complex associations between disease phenotypes and endotypes and better means for diagnosis and treatment of allergic diseases. However, there is still much to learn. Thank you very much.